Welcome back. Now, today we're going to do another biology BGCSE paper review, and we're reviewing 2017 paper 2. Remember to always write your school number, your candidate number, your surname, and initials. Remember also to read through the instructions carefully, and if you have any doubt or if something is not clear, please do ask questions before the examination start. Now let's go into the first question. This question states that the diagram shows a mangrove plant in its natural environment. Now you're going to observe the plant and the environment as well, and then jump into the questions. So let's look at the first part of the question, which is part A1. The question reads, that state the specific name of the type of mangrove shown in the diagram. So the first thing you're going to do is to go back to your diagram, observe this plant, and you notice the obvious things that are there in terms of a plant include the type of root system, the type of fruits that are on the plant itself, and you also will see the types of leaves. The leaves in this case, or the flower, they're not so clear, but you can see the root system, which is a good clue of what plant it is. So let's go into and answer this. And based on the rooting system, this plant here is called the red mangrove. And I'm going to write the scientific name as well, so you can always remember that. And since it is a red mangrove, this is called the Rhizophora mangel. The scientific name is written with the genus, and always start with an uppercase letter for the genus. And so this is Rhizophora mangel, and mangel is the species, and that is start with a lowercase letter. All right, and so that is the specific name for the red mangrove. Now, the other part of the question here, part 2 of A, it said give 2 reasons for your choice. And based on the plant, if you should go back to it, you will realize the 2 most obvious things that are there is the type of fruit that is on the plant and also the rooting system. Unfortunately, you cannot see the leaf so clearly. If you can make up the leaf, then that will also be a good choice as well. And so I'm going to write 2 possible options here. And so one of them is the type of fruits that are shown on the plant, and those hanging things with the root coming out of it, because the fruit is germinating on the plant. And so what those fruits are called, germinating fruits, they are called propagule. So you see propagule present, and the type of rooting system, you will see that the root will be prop roots. So these are the 2 things that are present on the plant to tell you that it is a red mangrove. Let's go to our another section of the question. And it said, using the diagram, give 1 example of a producer, a consumer, and a carnivore. If you look at the diagram, look at the animals present, or the organisms present, you can identify the producer. You will see some algae. You will also see seaweed. You see a shark. You see a crab. You see a starfish. You also see a fish, a turtle. You also see snail. We also see starfish. All right, so let's look at these options now. And so based on all of that, a producer could be the algae. And you can also, I'm going to put another option there, but again, you only need 1 based on the instruction, right? So always follow the instruction, but I'm going to give you more than 1, so at least you know when you're studying, you know what to do. So the seaweed is also another one, okay? For the consumer, which is the easiest part, actually can write any of the organism except the producer, because all the other organisms, they will be consumers. So the turtle is 1, of course. The crab is also 1, and again, the instructions say only list 1. So I'm just giving you a number of them so you can choose from that. The fish is there, you have the starfish, and so on, right? So the snail, starfish, etc., etc. All right, so you can always decide what you want to do. Choose 1. You cannot be wrong in this case. This is the easiest part. Now the carnivore, Now, is the carnivore is any organism that eat another animal. So animal eating animal, all right, or animal eating meat. So to be a carnivore, the two obvious one in the diagram there will be the shark. The shark will be one, and the fish also is a carnivore. All right, so the fish could also be another one because the bigger fish could eat smaller fish. All right, so those are the possible answers there. Now let's look at the other part of the question. And this part reads that draw a food chain of 4 trophic levels using organisms shown on the above diagram. And so if you go back to the diagram again, you will notice the type of organisms present. So observe it, look, and decide what you want to choose to make up your 4 trophic level. Now, what it means by trophic level is that you must have 4 organisms, including the producer. The producer is the first trophic level. The trophic level is the feeding level of the food chain. So let's start with the producer here. I'm going to have 4 organisms in this. So we can have here the algae. Algae is a producer. And let's put the other organism will be here. What could eat the algae is the snail. So the snail could eat the algae. And what could eat the snail could be the starfish. And what could eat the starfish could be the crab. All right, so that will be our food chain. And so remember now, to make this accurate, you need to also draw your arrows. The arrows will indicate what is eating what. Okay, so putting the arrows in. All right, and that's your food chain with the 4 trophic levels. All right, so that will be your answer right there. All right, so let's go to the other question. This part of the question reads that state the name of the secondary consumer in the food chain drawn above. So in this food chain, you're looking for your secondary consumer, which means is the organism that eating the primary consumer. The primary consumer here is the snail, so the secondary consumer is your starfish. Let's put our starfish here. So that will be answer. And in part 3, it says explain the effect, if any, on the food chain of removing the secondary consumer. So in other words, if you take out the starfish, so we'll look at this now carefully. If you should remove the starfish, let's put an X through the starfish. If you take the starfish out, right, there's nothing eating the snail. So the population of snail will increase and the algae will decrease, okay? So that's exactly what will happen here. So let's put that in as our answer. So the population of snail will increase. Let's put it right here. So the population of the snail, and the snail is the herbivore or the primary consumer. The population of snail will increase, resulting in a decrease in the algae. All right, so that's exactly what will happen there. All right, so as I take the starfish out, there's nothing eating the snail, so the snail will increase, and because the snail eating the algae will decrease. All right, so let's jump to another question. All right, and this question reads now that the diagram shows the human digestive system. Now we have some letters there, and you can look at the letters and actually know what they are, and so we can just run through it real quickly. And so here we'll have our esophagus, which is K. L will be the stomach, M will be the pancreas, N will be the small intestine, and Q and O will be the liver. All right, so let's now go to see what they're asking us to do. All right, so it's a name the structures labeled K, M, and N. So again, K is the esophagus. So let's write that in real quick. That's the esophagus. And M, if you go back to the diagram, M is your pancreas and N is the small intestine. So let's put those in real quickly. So here we have our pancreas and we have our small intestine for N. All right, so good. Now for part B of this question, it reads, it said draw a line to the gallbladder and label it X. So the gallbladder, which is the part that's storing bile, it will be here, and so we could label that X. All right, so that is your gallbladder. All right, so our next part of the question. Let's see. Get into that real quickly. All right, so I want to see how X is properly. Okay, great. All right, so now jump into the next part of the question. And next part of the question says name the substance stored in the gallbladder. And the gallbladder will store bile. And bile is produced by the liver. And bile is used to emulsify fat. In other words, break down fat into small droplets called fatty droplets. And now this part of the question is to complete the table below by matching the function with the correct letter of the part on the diagram. So the part that breaks down drugs and alcohol is the liver, and the liver is O. So you could put O right here. Produces insulin and amylase, and insulin is a hormone. Amylase is an enzyme. In fact, you have other enzymes produced by this structure, and this structure is called the pancreas. So this structure is M, and you could always go back to your diagram and look at this carefully, but by memory, we know the pancreas is M. And then it's at the site where absorption, and it's a matter of fact, it's a most absorption takes place. And this structure here will be the small intestine. The small intestine is where absorption takes place, and it is labeled N. In the large intestine, you have reabsorption of water taking place. All right, so let's put our N right here. N is the answer for this one. All right, so that will be your choices, O, M, N. Now we'll jump into the next question. It's a name the acid produced in L and state one function of this acid. So the L is the stomach. So go back to the diagram. L is the stomach. And matter of fact, once they mention producing acid, then you know it's the stomach. And so that is L. And so the acid that is produced there, it is called hydrochloric acid. Okay. So this is hydrochloric acid, or the symbol for it is HCl, but it's a name it. So write the name, always write the name. But I'm going to put the symbol for you as well, for you to also know it. It's going to be HCl. All right, but they ask you for the name, so always write what they ask you for. Please read the question properly. So this is hydrochloric acid. I put the acid part. All right, so hydrochloric acid, the symbol is HCl. The function. The hydrochloric acid has a lot of functions, but in terms of digestion right now, I'm going to put two functions for you. They only ask you for one function, but let me just give you two real quick. So one of the function here is to stimulate. So it stimulates the action of pepsin. It stimulates the action of pepsin. And pepsin will break down the protein. Okay, so it stimulates the action of pepsin. So that's one function there. Another function is that it also aids in killing bacteria. So the hydrochloric acid will also help to kill bacteria. So kills bacteria. All right. Kill bacteria that are entering the stomach. All right. So very importantly, that's the two main functions there of the hydrochloric acid within the stomach to stimulate the action of pepsin and also kills bacteria that are entering the stomach. All right, now let's jump into our next part of the question. All right, this question is completed. So another question here, and this is question number 3. And this one is saying that in 2010, 250 million tons of waste was collected before recycling. It said the statistic of the waste collected is shown in the table below. And so if you notice a number of things here, you will see that a label from A to H, and you have different types of waste collected. So you could go through the waste, and you also have the percentage of the waste collected. All right, so let's see what the question is saying about these waste substances. Now it says state the difference between the term biodegradable and non-biodegradable. So let's talk about biodegradable first. So biodegradable are substances. So biodegradable are substances that can be broken down by decomposers. All right, so they can be broken down by decomposers such as bacteria. Let's put that in, such as bacteria, right? Such as bacteria. All right. Now, while non-biodegradable, let me put that here. While non-biodegradable cannot be broken down by decomposers. All right, so that will be the answer there, all right? So the biodegradable can be broken down by decomposers, while non-biodegradable cannot be broken down by decomposers. And so the word bio means living, and degradable means to break down. So break down by living things. All right, so let's go to the other part of the question here. And they said, using the information from the table, calculate the mass of the metals that were collected. And so let's go back to see what the table is saying about the metals. And so remember now that the total waste collected was 250 million tons. The metals accounted for only 9%. Okay, so what we want to find now is 9% of 250 million. So let's go down into that. As metal is only 9%. So we're going to write our 250 million and find 9% of that. So we have our 250 million. All right, that's 250 million. I want to find 9% of that. So you can just multiply by 9%, which is simply 9 divided by 100. So, but 9%, and that will give us, just calculate that, is give us 22,500,000. Yes, that's correct. 500,000. All right, and that is 500,000 tons. All right, and so you can also write this a little bit short if you want. You can just say, if you want to write it in words, you can just say 22.5, so it'd be 22.5 million tons as well. Okay. All right, so that will be the answer right there. Okay, so let's put or right here. Okay. All right, so that will be answer, 9% of the total. That's the metals. And then it said, now using the letters A to H from a table, identify the waste which is non-biodegradable. So looking for the non-biodegradable substances, the substances that cannot be broken down by bacteria or decomposers. So paper and paperboard, they can. Food scraps, yes, they can be broken down, so they are not non-biodegradable. Those are biodegradable. Yard trimmings, those are good biodegradable substances. They are organic materials. Plastic and styrofoam, yes, that's a correct answer right there. So let's just stick this so we remember exactly what we are talking about here. So this is one of the answer here. E, metals, yes, that's non-biodegradable. F, what is saying, rubber, leather, and textile, yeah, that's non-biodegradable. and wood is biodegradable, and H glass, that is non-biodegradable. So it's going to be D, E, F, and H for our answers. All right, so let's go down to a chart and insert those real quickly. So one of them is D. Next one will be E. Other one will be F, and the last one, that was H. All right, so those will be your answers right there. All right, good. Now let's move on and go into another question right here. All right, so next question now reads that from garbage to garden is a popular composing slogan. It's a name one waste listed in a table that can be used for composting. And composting is when you decay organic materials and allow them to form rich soil called humus. And that can be used to add nutrients back to your soil. And so let's list some things based on what is present in the chart that can be used for composting. So a number of things that are there. So the yard trimmings can be used. The food scraps can also be used. And the wood can also be used. But wood won't be a good choice because it will take a longer time to break down. But it possibly could break down the wood in small pieces and then you can put them in your compost. But large wood will not be the good choice. It will take a long time. But again, you can make it faster by breaking it down, cut it up in small pieces, then use it. So food scrap is a excellent choice. So the food scraps. Yard trimmings is also good, but it's still list one, so I must give you the all of them, and so you can decide what you want if you should see a question like this. All right, and wood. So those are the possible choices. It's a give one reason for your choice in D1 above. So why would you choose any of these? And simple because these substances listed above, they are biodegradable. Okay, so they are biodegradable. All right, and since they are biodegradable, and so they are able to break down easily, to be broken down very easily by decomposers. All right, so they will be decomposed really fast and really easy. All right, so that will be your answer right there. Let's go to another part of the question. And this part reads that burning garbage at the New Providence landfill dump site has made national headlines. It said describe 2 environmental problems associated with burning garbage. And in fact, there are a number of problems that we might have, but they only asked for 2, but I'm going to give you at least one more. So let's start with the first choice, and it said describe, right? So notice this question said describe 2 environmental problems. So one could be air pollution. So let's put air pollution right here. All right, air pollution. All right. So air pollution is one. And let's talk about air pollution. So how this would happen is that harmful gases, right? Harmful gases can escape in the environment, right? Escaping in the environment. All right. And escaping the environment, it could cause some problems. So what can it cause is causing respiratory problem, right? So it causing respiratory problems. So let's put that real quick. As a person who living near that dump site can experience respiratory problems because of the harmful gases and so on. All right. Another possibility here that we can have is global warming. Okay. And you have a global warming, and how the burning will contribute to this, because the burning on the dump site will increase. So there'll be let me just write this real quick. An increase in greenhouse gases. All right, so an increase in greenhouse gases, and we can give some examples such as carbon dioxide, okay? Such as carbon dioxide, causing warmer temperature, right? So the temperature of the environment will increase, causing warmer temperatures. All right, great. So that's another possibility right there, right? We can also have more, again, such as we can also have acid rain, okay? And so the rain can become acidic. So we may have acid rain. I'm just going to put that there. And of course, just quickly just to explain that, is that the gases will mix with precipitation, water, which is the rainfall, the hail, and sleet. But of course, in New Providence, you only have rain, all right? So of course, the chemicals will mix with rainfall and will lead to the rainfall become acidic. And some of the gases that will cause this will be like sulfur dioxide, right? Nitrogen oxide. So here we could have acid rain because of acidic gases. So let me say acidic gases mixing with rainwater. All right, and some of those gases, as I mentioned, could be sulfur dioxide or nitrogen oxide. All right, and so mixes with rainwater or precipitate and cause acid rain. So I was put in there as another option. All right, let's move on to another question real quick. All right, so this here is another question here, and this one is saying, I'm just going to zoom into that a little bit so you can see a little bit better. It's an investigation was carried out with flowering plants to determine how many of each type of seed would germinate. It said 100 of each type of seed was planted, and the percentage of the seeds that germinated is shown in the graph below. So you planted 100 of each type of seed. So let's get the type of plants that were used here. And so we have a number of plants. We have hibiscus based on the graph. Hibiscus is over this side. We have the oleander, we have the morning glory, we have the corn, and then we have last over this side is the Porsman orchid. And so on the y-axis, we have the percentage of each seed that germinate, and on the x-axis, we have a type of plant. And so at the bottom of the graph, it asks us to define the term germination. So germination is when a young plant will grow from a seed. So it's a process. So let me just write that in real quick for you. So it's a process by which a young plant will grow from a seed. All right, so the process is a process by which a young plant grows from a seed. Okay, it grows from a seed into a self-sustaining plant. So that means the plant will be able now to photosynthesize. That's when germination actually ends. All right, so the process by which a young plant grows from a seed into a self-sustaining plant. All right, so that's what germination is. Now, here now it says state one environmental factor necessary for germination to occur. And there are a number of things needed for germination to occur. And so let's just talk about them real quick. Let's see if I could put in all of them. You must have available water. So one is, they put an asterisk there, so you're going to see them separately. So available water. Your correct temperature must be there. So you must have the correct temperature. You must put temperature. All right, you must have that. You must also have available oxygen. Notice here, there's no need for carbon dioxide. There's no need for light. Okay, so do not make that mistake. So again, we must have available oxygen. The plant needs oxygen for germination. Why? Because the plant will need to respire to produce energy to grow and develop. And Also, the pH is also important. The correct pH is also important. For some plants, they are pH sensitive, so yes. So the pH level can also affect germination. So I'm going to put here correct pH. So the correct pH, and pH is a lowercase p, uppercase h, okay? All right, so those are 4 major things that are needed for germination to occur. Again, remember, the plant do not need light, neither do the plant needs carbon dioxide for germination. Those are not needed. All right, let's go to the next part of the question. It said, which flowering plant has the lowest percentage of seed germination? You could just go back to the graph and see which one is the lowest here. And C is the lowest. So C, what is plant C? Let's go for plant C. The plant C is the morning glory. So the morning glory is the least to germinate. Let's go to that question. So this will be the morning glory. Quite easy to read from the graph, a matter of fact, right? The morning glory. Right. So we have that. So the next part of the question now, let's see what it's saying here. It said how many corn seeds germinated. And so let's go back into what is happening here. We can get this quite easy from the graph again. How many seeds of corn were germinated? So corn is D. So we're looking at D right here, which is this structure. So D will be here. All right. All right. Unfortunately, this might not be seen easily. But what I want to point out, you have 80, you have 100. So this number here must be 90. So I just want to point that out. Okay. So 90 is here. I'm going to help you out. So we have 1, 2, 3, 4, 5 lines in between, right? So if there are 5 lines in between, that means each line is 2. So let's count this, go up to 1, 2, 3, the 4th line. So this must be 88. Okay, so 88. So, but the thing is now, because this is in percentage, and I'm going to show an easy mathematical calculation here. If it's in percentage, and there's only 100 seeds that were planted, it therefore means the percentage will convert to the number of seeds. All right. But I'm also going to show the calculation for it as well. You can just put the number there, which is 88, but I will show you exactly how to calculate that. All right. So in terms of calculation, you'll want to find 88% of 100. All right. Then again, you don't need to go through all of this, because since there's 100 seeds and it's in percentages, convert exactly to percentage. So that will equal to 88 seeds. Okay. So 88 seeds of corn were germinated. All right. So next part here is to calculate the difference between the number of corn seeds which germinated compared to the number of Pursman orchid. And it says show your working. So first have a look at the graph. We already know that we have 88 seeds of corn that were germinated. So looking for Pursman orchid, and Pursman orchid is E. And again, if we go across here, this will be 50 right here. So let's put that in for you real quickly. So the number that is missing here will be 50. Okay, let me take my time and put that in. All right, just ignore that bad writing. I apologize for that. Yeah, but it's 50. And then this line here again is the 4th line. So this is 8. So this will be 48. Okay, so that is 48 of the Pursman orchid that were germinated. So the difference between that here will be 88 minus 48. So let's put that in here. So here we're going to have 88 minus 48, which will equal to 40. Okay, so 40 seeds. That's the difference between the corn and the Pursman orchid. All right, let's go to our next question here. It says state the function of a tester and the micropile in a seed. Now, the tester will have a number of functions, right? But the basic function for the tester is to protect the seed. All right, so to protect the seed. All right, and what is really protecting the seed is the embryo. Okay, so the embryo of the seed, really. So the embryo of the seed. And when it's protecting the embryo of the seed is especially, let me put this in, especially during dormancy. That means when the seed is not growing yet, when germination is not taking place, especially during dormancy. All right. That's especially when the tester is most needed for protection. When the seeds start to germinate, then the tester will break open and is not needed anymore. The micropile now, the purpose of the micropile now, it allows water to enter the plant, or into the seed, I should say, enter the seed. All right, and what the purpose of water entering the seed is to stimulate germination, right? So it allows water to enter. Never put that in. Water to enter the seed. All right. And so can just leave it as that. So water to enter the seed. Let me take this little piece out. Enter the seed. And what is needed to stimulate the process of germination, right? You can leave it as that. All right. If you want to put the part that says what is needed to stimulate germination, you can also add that as well. But it's only 2 mark questions, so that is sufficient. Now, this part here said draw diagrams showing the external features of a leaf of a monocotyledon and a dicotyledon. So let me draw the monocotyledon first. And so when you draw in monocotyledon, a matter of fact, I could draw anyone, it don't matter. But let me put them on the side so at least you will see. I'll just put the lines down instead of, to save some time. So I'll put monocot right here. So monocot will be a pretty much narrow leaf. So you're going to draw a narrow type of leaf, all right, for a monocot. And what is important about the monocot is that you're going to have some parallel leaf veins. That means the veins in the leaf will run side by side, right? So you're going to draw them like that. That's the most important thing. And what I suggest to you is to label this. When you draw a diagram, please annotate a diagram by just typing something or writing something, I should say. So this is the monocot leaf right here. And you want to label this to say, okay, narrow leaves, parallel leaf veins. Please do that when you draw, okay? It's always good for you. It's an advantage, right? And then for the dicot leaf, you're going to draw a relatively broad leaf. And what is also important for this leaf, you're going to draw the veins of the leaf being branching. So you're going to put branching leaf veins. And again, you can label that. I'm going to put like a few more just to save time, all right, because I don't want to keep you too long here. All right, I'm trying my best not to. All right, so yep. So that will be branching leaf veins, broad leaves, okay? And that will earn you your marks. Let's go into another question. All right, so let's go into question number 5. And this one says the diagram below shows the components of blood seen through a light microscope. We have structure G, H, and F. And those are blood components. And if you look at them carefully, you actually will know them. All right. There's one that is not labeled, and which is this one. Let me see. I could label it real fast for you. And so this is the one here. I'm going to put the label afterwards, right? But we know that G is red blood cell. F is the plasma. H will be white blood cells. All right. And we have our phagocyte, which is the irregular shaped nucleus, and our lymphocyte, which is regular shaped nucleus. And so here we'll have our platelets, okay? So let's put that right there. Platelets. And platelets are called scientifically thrombocytes. So this will be our thrombocytes. All right, so let's now go into the question itself. This is what the question is actually asking us to do. All right, so it's a label. You give it a label FGH. It's a name the blood components, and then after that, now it said main function. So put the main function of these components and name it, and then put the function. So let's go to F. Let's go back to see what F is. So F is the plasma. Awesome. So F will be the plasma. So I'm gonna put plasma here. I put the uppercase P. All right, so that's the plasma. And the function here for the plasma is pretty much to carry nutrients and hormones around the body. And it also takes waste from the cell as well. So it, let me just put that real quick. So this one carries, the plasma carries waste, carries nutrients, and hormones. Let's see if that could fit in. And hormones, right? All right, so it carry nutrients and hormones to the different parts of the body, and they take away waste from this and take away waste from the cells as well. So please, you could just add that in. All right, matter of fact, let me decrease the font size here, and so I could fit in everything. Okay, so it carries nutrients and hormones. Okay, and the next function here, it take waste away from the cell. So it takes waste from the cells. All right, so those are the two main functions there. So the main functions carry nutrients and hormones and also take waste from the cell. All right, in terms of now G, let's look at what G is. And G, let's go back to the diagram to see exactly what it is here. And so G is our red blood cells. Okay, so G is our red blood cells. So here is red blood cells. All right. All right, and red blood cells, they are specifically called erythrocytes. So let's put that in for you. So you know the scientific name for red blood cell is erythrocyte. Okay, let me just decrease this so it fit nicely. All right, so erythrocyte, and the function for erythrocyte is our red blood cell is to transport oxygen. Okay, so they transport oxygen. All right, so transport oxygen around the body to supply the cells with oxygen. And H, which is our white blood cells, that one we will know because based on what we have here, this is the white blood cells. The white blood cells, they are called leukocytes. All right, and the function of white blood cells is to fight infection. So they fight infections and diseases. All right, so that's the function of the white blood cells. All right, so good. All right, so again, you can just scroll back into your diagram and look and see exactly and so on. All right, so let's now go into another part of the question here. Now, this one is asking which labeled structure increases in number when there is an infection in the body. So there, even based on the function, once they get the function correct, you actually will know exactly what is causing this. And so this is structure H. They said which labeled structure. Did not ask for the name. It said which labeled structure, and that is H. All right, now we said name 2 types of blood vessels found in the human body. And I'm going to list all 3, but they say name 2, so you only name 2, okay? So here we have arteries. And we also have veins, okay? Oh, wow, I missed my R out of this. Arteries. And we also have veins, okay? So there are 3, in fact. So we have arteries. We have our veins, and we have capillaries. All right, but again, they only ask you for 2. Okay, so please follow the instructions. All right, but I'm listing all 3 here because you're using this for studying purposes. All right, so now part 2 of this question, it says state one structural difference between the 2 types of blood vessels named in B1 above. All right, so what are you going to do now? Easy way to do this. I'm going to give you more than one differences, right? And so let's just compare the veins and arteries. So what I will tell you to do in exam to make life easy and easier to mark and easier for you as well is simply just to draw a line down this thing here. Easy way to do it. And then you want to put here, you could put your arteries on this side. You're going to make life so easy. Okay? You put arteries there, and over this side, you put veins. All right? And this is going to make life, I'm telling you, really easy for the marker and also easier for you. So, one of the first difference you can have right here is that the arteries, all right, they have no valve, right? So, no valve. Arteries, no valve. Okay? Veins, they do have valves, so veins have valves. Okay. All right, so no valves present, and these are valves. And next difference here, and then we're comparing the same thing. Okay. So arteries, now they have narrow lumen. So the lumen here is narrow, and the lumen in veins will be wider. So these have wider lumen. Okay. Another difference here, I'm just giving the differences so you can at least know them. So this here have thicker wall. Thicker muscular wall. All right, let me just put it on this side. Not going to look too nice there. Thicker muscular walls. All right. And the veins now will have thinner muscular walls. All right. All right, let me put it here so you see it better. So thinner muscular wall. All right, so those are the general differences, right? All right, so again, you only ask for one. All right, so you get the marks easy, but you need to know all. So this is a very easy way to do this when you go in the exam. Just create a little chart and so you can use that now to show the difference between the two things quite easily. You can use a ruler so your thing will look nice and neat as well. All right. Let's go to our next question here. All right, again, this question says the diagram below shows the human male reproductive system. All right, we have some different structures here. And they said select the correct letter from the diagram which describe each of the following. And so it is asking you now a structure where sperms are made, and sperms, they are made in the testes. And so the testis is shown here, which is S. Okay, so testis is singular, testes is plural. Just remember that as well. Next one, say a structure which becomes erect during intercourse, and that is the penis. In fact, Q is the shaft of the penis, so let's just put Q right there. And that is filled with blood, because for it to become erect, it must be filled with blood, okay? A structure down which urine and sperm travel, that is the urethra, and that structure is P. And again, sperm and urine do not pass through at the same time, because there is a membrane that will separate the urine from coming out if you're ejaculating. All right, and so those are our letters there. It said, what is the function of the epididymis? The epididymis is this convoluted structure. So I'm just going to make a line there. That's the epididymis. That's a convoluted structure, or this coiled. You may want to describe it as being coiled as well. All right. And so the function of that is to temporarily store sperms until they are mature. So let me just quickly write that in. It temporarily stores sperm cells until they are matured. All right. So that's exactly what the epididymis is doing. They temporarily store sperm cells until they are mature. It's to explain why the structure labeled S must be held outside of the body. Let's see what S is. S is the testis. So the testis is not in the body. It is in the scrotal sac or in the scrotum, and it's outside of the body. And the very, very simple and basic reason for that is because sperm cells are produced at a lower temperature, slightly lower temperature than the body itself, okay? Let me write that in real quick for you. So this is because sperm cells are produced at lower temperatures, all right? And the temperature is only slightly lower, really, about 1 degree slower. Than the body, all right? So the temperature must be a little bit lower. And so in colder time, the testes will ascend closer to the body, become warmer. And in warmer time, the testes will descend and move away from the body and to become a little bit cooler, all right? So males will understand that because they would have experienced it if they notice it. Now, part D now said define fertilization. And fertilization is when the gametes come together and the nuclei will join. So this is simply saying that the union or the fusing or the joining, whichever word they want to use. All right, so this is the union of a male and a female gamete. So the gametes will join, they will meet, okay? And when we talk about here, we talk about fusing of sperms and egg, okay? So we can even say the fusing of sperm and egg. All right, so that's what it is. All right, now next question. And this one here now, it says suggest and explain or cutting the structure labeled R would affect the possibility of fertilization. So let's look at what R is. And R is the sperm duct or the vas deferens. So cutting the sperm duct, that means it cannot transport any sperm, right, outside the body. And so what will cause here, again, in terms of suggestion, the sperm not able to leave the body. So it's going to be simple as this. It's a no sperms will be present in semen. So let's put that as the effect, right? So no sperm, let's say no sperm cells will be present in the semen. All right, let's try and go as fast as possible. Therefore, no possible. So this will happen, no possible. Therefore, no possible fertilization. All right, so there's no possible fertilization taking place. All right, great. All right, so that's exactly what will happen here. No possible fertilization taking place at all. Now, here now it said name a method of contraception which also stops the spread of sexual transmitted diseases. And this one is the use of condom. That's the only one that will do that, all right? Use of condom. It prevents fertilization and also prevent STD. All right, use of condoms. All right. So let's go to another question. I think this should be, no, okay, but I think we have one more question. It should be 8 questions. Let me see something here. Question number 7. Okay, so we're almost there to the end. All right, so here now, with this, we're going to look at this now carefully. And I say the diagram below shows the internal structure of a leaf. Here we have structure Q. Okay, we have structure Q right there. That's the only structure they actually labeled. It says on the diagram, label a palisade cell and a stoma. So let's go up here now. The palisade cell is those long elongated cells that where the most chloroplasts can be found. And so here will be, let's just put a line there. And so one of these will be the palisade cell. The entire thing is a palisade mesophyll layer. So this is the palisade cell. Let's put that right there. Palisade cell. All right. And it also asks us to label the stoma as well. So let's label the stoma. And the stoma is the leaf pore. All right. So leaf pore or the opening in the leaves. All right. And that's the place where you have gaseous exchange taking place. So here is the stoma. All right, so that will be our structures there. Palisade and the stoma. Now this one here said draw a straight line from each structure to the function it carries out. So look at them carefully. We have the cuticle, the palisade, and we have the phloem. Is there anything else? No, we have one more. Yeah, there's like a box there. So let's match up these things. All right, so we're going to keep it right there and match them up. And say cuticle, what the purpose of cuticle? Cuticle is same as the waxy cuticle, right? And the waxy cuticle is what will reflect light and also make the leaf waterproof. So we see our answer right there for the cuticle. All right, so this will be this. All right, I'm going to kind of use different colors so we could kind of see them differently. All right, so let's look at another one here. Let's put that blue maybe. All right, palisade. Let's think about our palisade now. All right, palisade is responsible for photosynthesis. So in other words, it will help to produce glucose. So that will be our palisade right there. All right, let's look at another one again. All right, so let's look at a different color for this one. Probably use green. All right. And so let's look at the phloem. The phloem is what now is a transporting vessel that transport plant food such as sucrose. So this is straight across. Let's change this into something else. Let's put that to the blue. Okay, right. So let's carry it right over there. All right, so that is that for us. All right, so excellent. All right. So let's look at the last one now. And our last one, we are going to now look at the sponge, which is the last one. So definitely that will go straight up to that one. All right, let's put that into probably black. All right, so those will be our options. All right. So you should be all right with that. All right, we should be good. All right. All right, great. Excellent. All right, so you can see the matching there. Let's go to the next question or next part of the question here. Is to explain why it is important for the structural label Q to be transparent. And the structural label Q here, that is, those are, in fact, the Q right here, they are the upper epidermis of the plant. On top of it, you'll see the waxy cuticle. So Q, why is it transparent? Why those cells are transparent? And it's very simple. It's at the top of the leaf, right? So light have to go through it. So the purpose of this is to allow the passage of light to the photosynthetic cells. So I'm going to write that right there. To allow the passage of light. Okay, come on, light to the photosynthetic cells, okay? All right, and when we talk about the photosynthetic cells that I'm talking about here is the palisade mesophyll cells and also the spongy mesophyll cells. Those are the ones in the leaf that are responsible for photosynthesis. All right, so let's go now to the next part of the question here. All right, and this question now reads that here it said that the diagram shows the apparatus set up for an investigation into transpiration. A cut chute is attached to the photometer. So there's a photometer now, and notice the plant, it involves water and so on. And so water will be traveling through the tubing. All right, and so there's a beak of water, which is pretty much the reservoir, and like the reservoir, so they're going to suck water from here. The reservoir just add extra water into it to reset the marking or the bubble. There's an air bubble right here. All right. And so it's a define the term transpiration. And transpiration is the evaporation of water from the uppermost part of a plant. So this is the evaporation of water from the uppermost part of a plant. Uppermost part of the plant. All right, so that is transpiration. Now the question here now read the next question read, what does the photometer actually measures? Now the thing was saying that we're investigating transpiration, right? But unfortunately the photometer cannot measure the rate of transpiration. It does not measure it. I'm gonna tell you why. Because even though it is measuring the water going into the plant, not all the water that go into the plant will be transpired, right? Not all of it will be evaporated. Some of the water will be used for photosynthesis and other chemical functions, right? So the only thing that can be measured by the photometer is the water uptake by the plant. Okay, so the only thing being measured is the what? Water uptake, not transpiration. Okay, water uptake by the plant. That's the only thing it is measuring. Because again, some of the water will be used for photosynthesis and also be used for other chemical processes. All right. And so is to place an arrow on the diagram to show the direction in which the air bubble will move during the investigation. So the air bubble will be moving this way here, going towards the plant, because the reason for that, as air is being sucked up, right, or water being sucked up by the plant, then what will happen is that the bubble will also go in that direction as well, right? The bubble will also go in that direction. So that will be our direction of the movement of that bubble. All right, let's go to another part of the question here. It says state two changes in conditions that would cause the bubble to move faster. So in other words, what will cause the bubble to move faster is anything that will affect the uptake of water in the plant. That makes sense, right? Because the plant is taking up more water, means that the bubble will have to move faster because more water is taken up. All right. So the rate of the rate of which the bubble moves indicate how fast water is being taken up by the plant. So there are a number of factors here that could cause this. And technically, anything that affects transpiration or cause transpiration to increase is a factor, and also anything that also increases photosynthesis. So let's think about them. So we can have an increase in temperature. I'm going to give you a number of them. They say only two, but they said so increased temperature could be one. All right, because increased temperature, increased evaporation of water, right? You can also have here decreased humidity. So if humidity decrease, so decreased humidity. That's another one. I'm going to keep on listing. You can also have an increase in light intensity. So increased light intensity. All right. You can also have an increase in wind, because the more wind you have, all right, is the more water will move away from the plant, causing greater transpiration. And of course, one I'm going to label right here, which sometimes we forget, that can cause water to uptake by the photometer is the increase in photosynthesis. But what I want to add to this is also that the one I'm going to put here is also related to increase in light, because more light means more photosynthesis, right? But of course, it will be an option by itself if you did not have that. So increased photosynthesis. All right, so all of those will cause the bubble to move faster. Let me just put an asterisk so you see them separately. All right, so those will be your choices, or your possible choices. Anything that causes water to go fast in the plant is a factor. All right, so hopefully get that, hopefully understand that. Let's move on to our final question and we out of here. Now this question now states now that the question is about monohybrid inheritance and chromosome. Now part A say now, state the correct genetic term for each of the description below or stated below. So the outward appearance of an organism, which is things that you're seeing physically, that is called a phenotype. Okay. All right, so this is a phenotype. Let's put an uppercase P there. All right, and it's an organism with one dominant and one recessive allele for a specific characteristic. So if one allele is dominant and next one is recessive, this is heterozygous. Okay, so that is heterozygous. Okay. Another thing here is said that the genetic makeup of an organism responsible for its characteristics. So the genetic makeup here will be the genotype. All right, that's the specific makeup of the organism that determines the characteristics genotype. Because genotype will determine the phenotype, okay? And now you're saying now a section of a chromosome controlling a particular characteristic or trait. So the specific location there of the chromosome or section of a chromosome, that's called a gene. That's gene. Okay, that's a gene or genes, all right? Next part here now is said that an organism with either two dominant or two recessive alleles for a specific characteristic, and the term that we're going to use here is homozygous. Okay, so it's homozygous. All right. Now going further down here now, which seems like the last part. Yep. It said a pregnant woman wants to know what is the possibility of her baby being a male. So she wants to know what's the possibility of a baby being male, right? As you all know by previous knowledge, you're going to be 50-50, right? But let's do this. This is a parent's gametes organism, offspring. And so we have the male, the female. So what I'm going to do, I'm going to keep all the males on this side, all the things for the males on this side, all the things on the female about this side, right? And so the parents here, in terms of the chromosome, now for the male is X and Y. The female is only X. Now, the gametes coming from the male parent, it can either be some of the sperm cells will have X, while some will have what? Y. So it's either X or Y. Okay, so let's put our Y there. For the female, she can only give X. All right. And let's look at the offspring now in terms of the chromosomes. And I'm going to keep the male on this side. I mean, you don't have to, but based on the organization of this, I'm going to keep the males on this side. So to get a male, right, is the Y from the male merging with any of the X from the female, right? So since we have two X's, this Y could merge with any of those two. So right here we'll get our XY. Here we'll also get our XY. Again, I'm just keeping all the males on this side, all the things for the males on this side, all the things for the females on this side. You really don't have to, because you can just cross them by using your cross method, or you could do your Punnett square type of thing. And here we'll have the X from the male going to any of the X from the female. That will give you your female child. So two X's, two X's. So it's what is the possibility of her child being a boy? And her child being a boy is only 2 out of 4. So let's put 2 out of 4, which is a half. Or you can say 50% chance. So there's a 50% chance of having a male or a female child. All right, so this is the end of it. And again, I thank you for being here, and I hope this is helping you with your studies. Look out soon, very soon, for more reviews as we go towards the examination season. So have a great week. Take care, have fun, relax, study hard, do well.